Okay, good morning everyone, and welcome to the ELIXIR Czech Republic Friday Coffee, where we present the tools and services which is made for purpose of the ELIXIR Czech Republic infrastructure. Today we have Jiří Hon from Loschmidt Laboratories in Brno, and Jiří will tell us about the very useful tools they developed at the ICRC or Loschmidt Laboratories. It's about the EnzymeMiner and SoluProt, which is tools dedicated for the mining of the information about the enzyme. So Jiří, please, the floor is yours.
So thank you very much for a kind introduction. Today I would like to present you the two tools, EnzymeMiner and SoluProt, and my presentation will be divided into 4 sections. So I will start with the first section about EnzymeMiner. EnzymeMiner is motivated by the fact that millions of new protein sequences are added to public databases every year, thanks to fast and cheap sequencing technology. The problem is that we don't know the properties of most proteins because the required laboratory work to do that is time-consuming and expensive. But many of these proteins might be undiscovered enzymes that could be used as biocatalysts in industry or academia. Luckily, we can use computers here to mine soluble enzymes from the databases, but we need to solve several problems. First, we need how to distinguish enzymes with similar function from non-related proteins. And the second problem is how to select several tens or several hits for experimental characterization from thousands of hits. We solve this by developing EnzymeMiner, which is iterative computational pipeline that combines existing software like USearch, BLAST, Clustal Omega, or Cytoscape. And we use sequence similarity and presence of essential functional residues to filter the hits and to find only those that are relevant, that are probably enzymes with the desired function. The workflow looks like this. We have 2 required inputs, the query sequence or query sequences, and then the description of essential residues. And there is also one optional input, we call it other sequences. The first step in the pipeline is PsyBlast against NCBI non-redundant database. And then we do 2 filters. One is pair filter, which basically aligns the hit with essential residue template and checks the presence of essential residues. And also there is a next step, MSA filter. We construct MSA for all the hits and check if the essential residues are correct also in the MSA. Then we have a set of putative hits, which then go to annotation steps and generation of sequence similarity network. In the annotation, we integrate data from PFAM database using the InterProScan tool. And we also use annotation of transmembrane regions and data from bioproject and NCBI taxonomy. The sequence similarity networks are generated using Cytoscape and MMSEX2. So I think it's the right point to show you the user interface, how it actually looks, so you have a better idea of what is going on. So when you go to the EnzymeMiner homepage, you have 2 options how to enter the input. The required input is again the query sequence and the description of the essential residues. So the first easier option is to just use what we have in SwissProt. So for example, I will search for the haloalkane dehalogenase enzyme family, and I can choose some candidate sequence, for example this one. I think this is the LINB haloalkane dehalogenase. Yes, that's correct. And I see here that the LINB sequence has 5 essential residues. And I see also a plot where the residues are, and also I see annotations from PFAM, which domains are in the sequence. On the right side of the input, there is a sequence similarity network, which is generated for this enzyme family. Each dot in the plot is one sequence here, and the length of the edges is proportional to the sequence similarity between those two sequences. So those nodes that are more far away are more dissimilar by the sequence. When there is no edge between two nodes, it means that the similarity between those two sequences is so low that we disregarded their similarity. There is some similarity threshold applied. So when you have it selected, you can just name your job, enter your email, because the calculation takes about 5 hours or 3 to 5 hours. So we will be notified by email that the job is ready. The other option is this one. You can enter manually FASTA sequence for each query. You can also manually provide the description of the essential residues. Here I will explain what the essential residue is. It's actually a list of positions and specific amino acids which should be at that positions. So for example, for this enzyme family, we know that there is a nucleophile at the position 108, and it should be this type of amino acid. We can set up different amino acids here, which should be on that position. I can click here, load example, and you can see an example where we have two queries. Then again, I will enter my details and click next. This is example job, so actually this particular input is pre-calculated, so we already have results for that. So I will go directly to the results page. And the result of EnzymeMiner, there are two main sections. The first one is a target selection table, which contains all the hits from the mining in the database. And for each sequence, there is one row and many columns which annotates the hit. The sequence, the table has several sheets, like in Excel, and each sheet focuses on different type of annotation. So, for example, this sheet full data set is like listing just all the results. Then the next sheet, extra domain, is listing only those hits that has some extra domain additional to this primary domain. So we know that haloalkane dehalogenases has just this primary domain, and we see that we have hits which has, for example, GFP domain, some extra GFP domain, and so on. Then we have a sheet which prioritizes the annotation of a source organism and also the annotation of the temperature range of the source organism. So we can see hits which comes from organisms which are, for example, thermophilic. So these might be interesting targets for our characterization because probably these enzymes could be more thermostable, but it doesn't have to be always true. And then there is the sheet focused on the salinity, biotic relationship of the source organism, disease relationships, and also there are sequences which have a detected transmembrane region, sequences with a 3D structure in a PDB, and there are two special sheets. The first one is sheet which contains just the selected sequences. So you have a nice overview of what you have selected. And there is a special sheet which contains hits which you selected from the sequence similarity network. I will show you down below. So we also generate this type of visualization for all the hits. This is not the same like I showed you for the input of EnzymeMiner. Here, each dot represents not just one sequence, but many sequences. There is a sequence identity threshold. Here it's 50%. So all the sequences that share 50% or more identity are consolidated in a single node. So when you click on the node, the sequences that are in the node will be here, populated here in the network tab. So you can easily investigate which sequences are very like outlier sequences and you can enrich your selection so you cover the full sequence space of the enzyme family. So you just not select many hits from one node which contains only very similar sequences. So that's from the basic. That's quite that's all. And there is one annotation in EnzymeMiner which is particularly important, and that's the predicted solubility, predicted soluble expression of the sequence in Escherichia coli. This value is predicted by the second tool, SoluProt. So I will go back to my presentation. So, and the motivation for SoluProt is quite easy, because we know that soluble expression is a major bottleneck in heterologous production of many proteins, and it's hindering experimental characterization of enzymes found, for example, by EnzymeMiner, or found by UniProt web interface or NCBI protein database, and so on. So the problem is how to distinguish solubly expressible protein from insoluble one. And the problem is hard here because we just have very noisy experimental solubility data. It's actually quite hard to find data sets which were measured by the same method. So we just have a rough binary data if the sequence was successfully expressed in soluble form or not. And the solution here is that we created a machine learning model based on gradient boosting machine. We used a training set. We compiled more than 10,000 sequences from target track database, and we benchmarked our tool on independent test set against the existing tools. The accuracy of the tool is not like high, it's pretty low, but I will show later that it's still useful. So SoluProt is best of the available tools with area under the Rotz curve of 0.62 and accuracy of 58.5%. But here I will show you that for a prioritization task for which we use SoluProt in EnzymeMiner, the SoluProt prediction can give you some advantage, can increase the success rate of soluble expression in the lab. Here is an example. Imagine you have a set of 1,000 hits from EnzymeMiner, and you predict the soluble expression by SoluProt for all of the hits. And then you will remove 90% of the worst hits. So you will get 100 best sequences, best soluble sequences. And here we show that when you select and characterize those 100 sequences, you will in the end get 50% more, you might get 50% more soluble hits than in comparison to the situation you would choose the 100 hits just randomly. This experiment, you see this graph here, was done on the independent test set, which contains actually 3,100 sequences, and it's balanced set. So half of the sequences are soluble, half of the sequences are insoluble. So that's all for SoluProt. I would just show you the web interface. It's very, very easy. The only input is a FASTA sequence. Protein sequence is in FASTA format, actually. So I will click here to load the example so you can see how it looks. And then you just click submit, and the only thing you will get is a solubility value for each of the protein. This value is the probability of soluble class. So the higher this value is, the higher the probability of soluble expression. So you can also download the results in a CSV file and process it in Excel. So that's all for EnzymeMiner and SoluProt tools. Now I would like to thank especially to Loschmidt Laboratories, the great team. So they help a lot with development of EnzymeMiner and SoluProt. I would like to thank ICRC of St. Anne's University Hospital Also, ELIXIR Czech Republic and Metacentrum organizations for providing the computational resources for this project, especially EnzymeMiner, would not be possible without the support of computational resources, because each job takes about 30 CPU hours to calculate. So we could not provide, without the resources, this service to the public community, to everyone. And I would like to also advertise our portal. At this address, you will see all the tools we developed and maintain in Loschmidt laboratories, and you will they are categorized by the target use case. So if you want to design, or if you want to do other tasks with, for example, to fit calorimetry data and other stuff, so you have a right tool to use from our laboratory. So to summarize, I spoke about EnzymeMiner, which is a web server for mining of soluble enzymes and selection of promising targets. Then I talked about SoluProt, which is a predictor of soluble expression in Escherichia coli, which is integrated in the EnzymeMiner tool. Both tools together have a strong applicability prospects for protein engineering and biocatalysis. So using this tool, better biocatalysts might be revealed. I think it's quite a hot topic right now, because many pharmaceutical companies are trying to transform their production pipelines to biocatalysis, because it's perceived as more ecological solution. And those tools are freely available and stable software, I could say, with long-term support. I think this is quite important in bioinformatics community, that some tools have long-term support. And we also organize international training courses. So we are in contact with the actual users, and we have a lot of feedback from them. So, and the tools are widely used, with thousands of jobs calculated. So I think that's all from my side. Thank you very much for your attention, and if you have any questions, please ask.
Okay, Jiří, thank you very much for a very interesting seminar. And we have a few questions. So the first is, how often, if at all, is the EnzymeMiner used for analysis coming from metagenomic studies?
For analysis coming from metagenomic studies, you mean if EnzymeMiner is used to mine enzymes in metagenomic data?
Yeah.
Is it correct? It's not used because the database, metagenomic database, is not yet supported. We only search proteins in the NCBI non-redundant database. But currently, I'm working on implementing the EBI metagenomic database of proteins. I think it's mGenify database, to integrate it into EnzymeMiner. And I already have some preliminary results, so it should be done in near future. So then you could choose if you want to search your proteins in NCBI NR database or in the metagenomic protein database. So it's coming.
Second question is, from which source do you take annotations in the results table?
Yes, it's listed here. We take the annotations from NCBI Bioproject database, NCBI taxonomy, and from Interproscan. So basically, it's PFAM hidden inside there.
Okay,
and
so it's combination.
Yeah, so
okay,
it's combination.
Marianne has a question. Can it find enzymes with an activity that has not been described so far? And how many new enzymes activities are yet to be discovered? What do you think?
That's a good question. I think that it can discover enzyme with unexpected activity, but I would say that it will be by chance. So it's you cannot, like, I think it's impossible to provide such input that would like intentionally will be focused on finding new activities, because the input is based on the fact that you will provide a sequence, with the description of the essential residues, so you have previous knowledge on the system. So you know that these residues should be there. They have a specific function. They are important. So you can find enzyme with novel activity. We actually did 2 experimental studies. We ran a similar pipeline to EnzymeMiner twice and selected hits and experimentally characterized them in laboratory. And we found like hits with, for example, especially high activity, or some enzymes that had promiscuous activity. So they didn't do only the thing we wanted, but they did also something else. And to the second question, if there are some novel activities to be discovered, my personal opinion is that we still have something to discover. So I hope so.
Yeah, thank you very much. I don't see any other questions. So Izzy, thank you very much again. And please don't forget that next Friday we have another lecture in the series. So have a good day and see you next Friday. Bye.
Goodbye.